Hello, everyone. Thank you for joining us for the May North American Neuromodulation Journal Club. For attendees who are listening us for the first time, I would like to say a few words about this journal club and people who are behind of this initiative. The overall goal of the journal club is to bring together neuromodulation experts and experts from other fields with the interest in neuromodulation. We would like to concentrate on trainees and give them opportunity to present their newly published papers. This journal club already become an excellent platform for collaboration between basic scientists, engineers, and clinicians. Today's paper is an excellent example of that kind of successful collaboration. Our scientific committee is led by Professor Julie Pilitzis, who is the dean at Charles Smith College of Medicine and the Florida Atlantic University. I am a director of this journal club. I'm grateful for the help of our NANS coordinators, Adriana Zibinella, Lily Deschaff, and Kyla Gustafson. This journal class would not be possible without their ongoing help. Before we start, I would like to remind you a few rules of our webinar today. All of us are going to be muted during the presentation. Please write your question in the question panel on the left side of the screen, and we would be happy to address them in the end of the presentation. You can also find the file of this paper today on the left side of the screen. Webinar recording will be available on our NANS website in 48 hours after the presentation. Today, we will discuss how human brain mapping with multi-thousand-channel platinum nanogrids resolves spatiotemporal dynamics. I'm so delighted to introduce the first author of this paper and our speaker today, Dr. Yogbin Cho. Dr. Cho received his Bachelor in Science and PhD in Physics from Seoul National University in 2018. His PhD thesis was about hybrid dimensional quantum nanomaterials and nanodevices. Since 2018, Dr. Cho has been a postdoctoral scientist in Professor Shadi Daye lab in the electric and computer engineering at UC San Diego. Based on Dr. Cho expertise in nanodevices and semiconductor optoelectronics, his postdoctoral research in Professor Daye lab has focused on projects in the areas of novel development of brain recordings and display systems. Dr. Cho is a recipient of numerous awards. He was a post-Khadija Park Foundation Science Fellow in Physics from 2014 to 2017 and was awarded with the Best PhD Thesis Award in Physics from College of Natural Science, Seoul National University in 2018. In 2019, Dr. Cho was awarded the Individual Research in Basic Science and Engineering Grant from National Research Foundation of Korea. Throughout his career, he published 34 papers and co-authored 4 patents. Dr. Cho, we are so excited to have you today. Without any further ado, please take away.
Thank you very much. So thank you very much, Dr. Duff, for the introduction. My name is Youngbin. I'm very honored to have the opportunity to present my research in the NANS Journal Club. And I appreciate everyone for taking the time to attend this webinar. Today, I'll be presenting our recent paper titled Human Brain Mapping with Multi-Thousand-Channel Platinum Nanorod Grids Resolves Spatiotemporal Dynamics. This paper reports about the first human brain recording with multi-thousand-channel neurophysiological sensory grids. Before I start talking about the research, I would like to acknowledge the fantastic team where each person's expertise contributed to the realization of this work. Professor Shadi Daye at UC San Diego and Dr. Ahmed Raslan in Oregon Health and Science University led this nationwide team composed of excellent engineers, neurosurgeons, and medical researchers. Dr. Angelique Pop at MGH, who also presented last time in the NANS Journal Club, also provided many valuable insights into this work and help on the data visualization. Notably, the recent neurotechnological innovation we are making is a result of many years of close collaboration between Professor Daye and Dr. Raslan. Over many years, the collaboration between the team has implemented design improvements of the electrodes geared specifically for clinical use. This collaboration also resulted in a new NIH grant that will further accelerate the advancement of the new neurotechnologies. Additionally, a startup called Precision Neurotech was co-founded between them with a goal of getting higher resolution ECOG-grids approved for clinical use. I would first like to introduce the recent bloom of neurotechnology across the globe. Many neurotech startups have recently emerged to revolutionize the brain-machine interface, to improve the neurosurgical outcome, and to restore people from neurological diseases. All these companies are focusing on the implantable electrode technology for the human brain. Then why are all these neurotech companies focusing on the electrical recording of the brain? I will explain this in the next slide. The human brain is an extremely sophisticated and complex system that consists of 100 billion neurons, which is an astronomical number comparable to the number of stars in our galaxy. The brain consists of cortical columns that are basic processing unit of brain function. And then we could see that the cortical column is composed of interconnected neurons. The neuron as a basic building block of brain works fundamentally by electrochemical process happening at the cell membrane. Considering this electrochemical nature of the brain, I would like to emphasize that the electrical interrogation of the brain and neuron is the most direct way to interface with the brain. However, compared to the complexity of the human brain, the available electrical recording tools, especially for the human brain, are in its primitive stage. As we all know, in the history of science, new scientific tools have always led to new scientific discoveries. For example, the invention of telescope led to numerous discoveries that form our modern perception of the universe. Likewise, the new electrophysiological tool will enable new discoveries from extremely complex human brains. Today, I will share with you the result from the highest resolution electrophysiological tool ever applied to the human brain and the new discoveries we made with this. With regards to electrocortical graphy grids, in short, ECOG-grids, this electrode shown here is the gold standard in recording the brain activity from the human brain. But as you can see, the inter-electrode spacing is as large as 1 centimeter, and the size of each recording sites are too big to measure the localized activity of the brain. Thinking of the complexity of the human brain, this coarse clinical grid seriously undersamples and averages out the human brain activity. To solve this problem, many research groups, both in academia and industry, were proposing new technologies. But as you can see in these images, the size of the electrodes are too small for the human brain recording, especially for the intraoperative mapping. And one of the major bottlenecks of all these approaches were that they relied on 4-inch wafer process that limited the size of the electrode. More recently, a group of researchers reported on assembled electrodes that is basically a combination of multiple smaller grids to increase the spatial coverage and channel count. However, the gluing process increases the overall thickness, and these assembled grids generally face challenges in sterilization process for the human use. For these reasons, even until now, none of these technologies succeeded in recording human brain with thousands of channels. For the first time in the world, we achieved the multi-thousand-channel recording of the human brain during the awake craniotomy. The electrode we developed was conformal and compliant to the constantly moving brain, which allowed highly reliable brain recording. I will now show how we achieved this. For the translation of the biomedical devices to the intraoperative use, we need to account for the clinical setting. In the operating room, the patient undergoing the brain tumor resection is in the sterile field, while all the other electronics for data acquisition are on the non-sterile field. Considering the large exposure size of the brain and the large separation between the brain and the electronics, a large-scale electrode technology is essential. Additionally, human brain surface consists of complex 3D structure, which constantly moves. This calls for electrotechnology that are conformal and compliant to the movement of the brain surface. Moreover, in the operating room, we are seriously limited in time and space, so the electrode setup should be quick, reliable, and compact, all at the same time. My research covers all of these aspects. To meet all these requirements, we developed a large and scalable manufacturing process on a large glass panel. The electrode we could make with this platform is big enough to cover the entire exposed surface of the brain. And at the same time, it was long enough to bridge the brain surface to the data acquisition electronics. Although this is the largest microelectrode built in the field, it is also one of the most sophisticated electrodes that consists of nanoscale features such as platinum nanorods for high electrochemical sensitivity. The drill holes on the electrode was also critical to have highly reliable recording, as excess cortical fluid were perfused out through these holes, which enabled the electrode to be in high proximity with the brain. Additionally, the electrode's dimension and spacing were easily reconfigurable depending on the target cortical region we are interested in recording from. We fabricated the first 2 grids for human brain's motor sensory mapping, the third grid for the human language mapping in superior temporal gyrus, and the last grid with 0.15 millimeter pitch for rat brain spiral cortex recording. Another critical technical challenge was on how to connect thousands of channels quickly and reliably in the operating room. We focused on this solution. We found this solution in the CPU connector technology that was optimized over decades in computer industry. This large-scale electrode, together with the compact one-touch CPU connector, enabled the world's first multi-thousand-channel human brain recording. With these reliable macro fabrication process and connectorization strategies, we achieved over 99% good channel yield for 1,000-channel and 2,000-channel electrodes. To test the broadband and high-resolution recording capability of the platinum nanorod grids, we first mapped the primary somatosensory cortex of anesthetized rats before we bring this technology to the human. We consistently observed the clear evoke responses for whisker stimulation. The raw waveforms exhibited localized and high amplitude response as large as 500 microvolts, and the peak responses were observed about 30 milliseconds after the onset of the air puff of the whisker. We plotted the root mean square potential mapping of the evoke response, filtered at different frequency bands and as shown in here, and as expected, the spatial localization of individual stimuli was best represented in the gamma bands. This is because the high gamma activity is known to be highly correlated to the location and timing of the cortical activation, with a strong link to the spiking activities. We overlaid the localized high gamma activity responses to different stimuli and compared it with the anatomical structure of the rat brain. The localized high gamma activity responses to whisker stimuli agreed remarkably well to the anatomical structure of the barrels, as well as the homunculus legal position of the forelimb, hind limb, neck, trunk, and tail. These results show that the single grid-based mapping provides a reliable functional mapping of the rat brain with a superior resolution to identify the sub-millimeter cortical column. I would now like to show the human brain recording and some of the new discoveries we made with this technology. In the neurosurgery, the central focus, which is the boundary between the motor and sensory cortices, is an important surgical landmark. The central focus is conventionally identified by peripheral nerve stimulation and observing the polarity inversion point of the evoke potentials using the clinical grids. Our multi-thousand-channel electrode recorded extremely detailed feature of these evoke potentials at 100 times improved spatial resolution and 50 times larger amplitude. With this enhancement, we were able to find the true functional boundary in the 2D map. We also observed that the central focus where the brain folds was not aligned with the actual functional boundary. And this difference was as large as a centimeter. For the surgical purpose, defining the functional boundary was important. On the other hand, for the brain-machine interface, it is important to correlate the sensory and motor activity with the brain's activity. With the patient's individual fingertip being stimulated with the vibrotactile stimulators, the cortical responses were mapped on the brain. Vibrotactile stimulation of each fingertip evokes spatially distinctive high gamma activity patterns. After superimposing high gamma activity on an optical image of the implanted platinum nanorod grid, we could observe the fine spatial distribution of the neural correlates of vibrotactile stimulation. The sensory map of fingertips was all within the sensory cortex, which was in good agreement with the motor and sensory functional boundary we identified in the previous slide. Next, we asked the patient to move their hand. We observed distinctive high gamma activity evoked during the hand motion, where high gamma activity showed up first in the motor cortex and moved to the sensory cortex as the hand grabbing motion was completed. On the other hand, the beta waves showed high amplitude in the motion planning stage, attenuated during the execution of the motion, and increased once again after the motion was completed. And yeah, let me show the beta wave. These beta waves showed interesting propagating characteristics between the motor and sensory cortices. We analyzed the propagating characteristics of the beta wave by employing the phase gradient analysis that is originally developed by Dr. Lyle Mueller. This methodology is based on the principle used in the fluid dynamics field. In the preparation stage of the motion, we observed noticeable long-range beta wave propagating from the sensory cortex to the motor cortex. During the motion, the beta waves were suppressed and exhibited lack of coherence in the vector field. After the motion was completed, the propagating direction reversed as represented by the streamline. The propagation dynamic analysis of beta waves done with the large coverage, high-resolution platinum nanorod grids revealed the sophisticated spatiotemporal dynamics of the brain surface activity. Coming back to the neurosurgical aspect, from the epilepsy patient, we observed spontaneous, repetitive epileptic discharges. The brain mapping and propagating dynamics analysis identified the onset zone of the epileptiform activity and the pattern of the spread. To further identify the epileptogenic zone, neurosurgeons use cortical stimulator to evoke epileptic after discharges. Our electrode was configured to have multiple large holes, which enabled the bipolar stimulator to directly stimulate the brain tissue through the grid. We observe a clear after discharges and identify the propagating characteristics of the epileptiform after discharges. Our research on human brain sensory grids created lots of new research topics and collaboration, which includes human language cortex mapping, decoding a numerical process from the human brain, and redefining our understanding on epilepsy, and human spinal cord recording. This work also provided a solid foundation for our research group to be awarded with an NIH grant to improve the epilepsy treatment. In summary, our studies demonstrated the range of utilities of platinum nanorod grids for high spatiotemporal recording of neural activity for research and clinical use. Platinum nanorod grids use scalable and reliable microfabrication process to produce reconfigurable multi-thousand-channel sensory grid with a large coverage and high spatial resolution. In the clinical setting, the platinum nanorod grids were easily integrated within the operating room and allowed high fidelity broadband sensory mapping and pathologic epileptiform activity from the surface of the brain. And it also detected novel spatiotemporal dynamics of the human brain somatosensory activities. To conclude, platinum nanorod grid hold great promise in superior mapping during neurosurgery owing to its high spatial resolution and coverage. While maintaining excellent broadband temporal resolution. This technology has the capacity to scale to several thousands of channels, and we project that this technology could pave the way for a new era of enhanced neurosurgical mapping strategies. With this new technology, we are seeing new possibilities in therapies, brain-computer interfaces, and better patient outcomes. As a future direction of this technology, we are trying to integrate PtNRGrids with the implantable and wireless electronics for semi-chronic and chronic use, so that the next step will be the clinical trial for people with treatment-resistant epilepsy. This research is being supported by the NIH UG3 grant to accelerate the translation of the PtNRGrids for broader clinical use. UC San Diego is also establishing a current good manufacturing practice, CGMP laboratory, co-led by Professor Dye and Dr. Alexander Kalisi. This CGMP will regulate the manufacture of these grids to obtain FDA approvals for use in small clinical trials. To further leverage the utility of this 1,000-channel human brain recording platform in the operating room, we are also building a real-time visualization system with flexible micro light emitting diodes that will directly show the brain activities on the surface of the brain. NIH BP2 grant is supporting this research, and we already built the prototype device and testing it on the animals. Lastly, we are also investigating the questions on what will be the highest resolution ECOG grid that is really needed. Our most recent technology allows 1,000-channel PtNRGrids to have a pitch between the recording sites to be as small as 31 micrometer. So we hope this ultra high resolution ECOG grid will be able to answer this question. Thank you very much for your attention, and please feel free to ask any questions.
Thank you so much, Dr. Cho, for this exciting presentation. It's just unbelievable technology. It gives opening up so many opportunities and has so many applications. Please write your question in the question panel. While we're waiting for questions, let me ask you some of mine.
Yeah.
So let's start from beginning. There are other grids on the market. So why nanorods? So what is unique about them?
Okay, so there are many like microelectrode technology that is recently being developed, but to have a very localized and high amplitude signal that is specific to that functional region, we need a very tiny but a very low impedance channel. And we found out the solution from the PtNRGrids that has despite the recording site area being 30 micrometer, because of the nanoscale 3D geometry, it allows a very large surface area, so that it has a very low and very sensitive recording capability. The other alternatives are like PDOT, PSS, that have a different sensing mechanism. It's a volumetric sensing based on the molecular chain. And we believe that for our recording and potentially for stimulation, PtNRGrids, that's a novel metal, composed of novel metal, would have could be a superior solution both for the recording and stimulation.
So to summarize, nanorods gives us ability for the higher amplitude, so they're most sensitive. Is that correct? So that
yeah, with the smallest size, we could have a very high sensitivity. And the other advantage I could mention about the PtNRGrids process is the manufacturing process is based on the vacuum physical deposition over a large area. So even if we scale the electrode size and the channel count up to whatever, we could still have a very uniform, like thousands of channels. The other method for PDOT deposition, they mostly rely on the electrode deposition of individual channel one at a time, and there can be some compromise on the uniformity, and also, as I mentioned, some mechanical stability.
Yeah, so our journal club is a part of educational committee. So I want to be a little bit educational here. So
yeah.
we need a higher amplitude to have a stronger signal. Yeah. So why low impedance? Why do we need? Why it's so important to have a low impedance?
So yeah, the low impedance will ensure the high signal to noise ratio. So with a low electrochemical interface, yeah, we could, as I first emphasized in the first few slides about the electrochemical interface, it will have the it can capture the electrochemical process of the nearby tissues most effectively, and the low impedance across the broadband of the all brain waves, we could capture some very low frequency local field potentials to high gamma activity to even some spiking activity frequencies. So it's the low impedance in a localized site to ensure the low noise and high signal to noise ratio.
Yeah. For you as an engineer, interface is very important. For me, low impedance, that means I can get, like you said, low frequency local field potential as an electrophysiologist. So yeah, it's important. And how deep? So it's sitting on the surface of the brain. So how deep this roads actually? I mean, how deep does it cover the whole thickness of the cortex? Does it go beyond? So how deep it is, or it's really sitting on the surface?
Okay, yeah. So this electrode is designed to be on the surface-most layer, so it doesn't get into the depths of the brain. But usually the craniotomy size is like 6 centimeter by 6 centimeter in the awake craniotomy, and we could fabricate the electrode as large as 8 centimeter by 8 centimeter, and some area between the skull and there's some area between the skull and the brain, and we could even slide it underneath the skull. So we could achieve like maximum of 8 centimeter by 8 centimeter coverage.
So it's actually has a very big spatial coverage compared to other electrodes, but it's not. Are you planning to stay that way, or develop and have a deeper way?
So you mean
how development of this technology is going to go in that matter? Is going to be that superficial, or you're planning to? Is it possible for that particular technology to cover deeper areas? Or it's not designed that way in the beginning.
Okay, so for this electrode technology, it is initially focusing on the intraoperative mapping with the surface most layer, and we'll like keep advancing this technology specific for the superficial layer. On the there are other like type of neural probes that is also being developed in Professor Shadi Diet's lab. So some is like some of them are like analogous to the UTGrid that has a penetrating probe to get a little bit deeper inside the brain structure and to capture the spiking activities there. And we are also developing a neural probe like analogous to the SEEG probe that could go like as deep as 10 centimeter deep into the brain. Yeah, so yeah, we are developing this technology will keep advancing into the surface grid and targeting for the semi-chronic and chronic uses.
Yeah. We have a question from Richard North. Are you able to record single unit activity? 31 micro pitch should allow this.
Okay. Yeah, so I would say that with our human brain recording, we haven't observed some spiking activity from the surface grid. We think that it is because of the relatively thicker layer of the PI matter between the grid and the surface of the brain. On the other hand, we also did some brain recording with this 30 micrometer planet nanorod grids, much smaller grids on the bird's brain, and we observe a spiking activity from this. So it is essentially capable of detecting the spiking activity, but for some animals or for some brain region that has a relatively thinner PI matter. So if we can ensure the proximity of the planet nanorod grid to the like spiking neuron, then we can essentially capture the single unit and the spiking activity.
So it's, to my understanding, and please correct me, so it should go deeper for the single unit recording.
Yeah, but our focus here is not on detecting the spiking activity. The other type of grids that we are developing, like something that looks like a flexible UCA grid, it will be doing that role.
Yeah, probably my next question will be in relation to this one. So you did a validation on rats, and yeah, you get fabulous results. But we have another kind of electrodes on the market, NeuroPixels. They are mostly on the neuroscience side in animal research. So I think it would be interesting to understand the differences. How different they are.
Okay. Yeah. So yeah, NeuroPixel is a amazing like depth probe that is one of the best in the field, and it is basically a CMOS integrated, like about 1 centimeter long and very narrow, like 70 micrometer wide, very narrow probes with like hundreds of channels, and it has a superior capability to record across the different layers and depths of the brain structure. However, we are the planet nanorod grid is not targeting for the same like it is not designed to record across different depths, but it is rather to map the functional boundaries on the larger area, like on the 2D map. The planet nanorod grid is designed to map the large area 2D mapping and the propagating wave dynamics. So I would say that NeuroPixel could be used to answer many neuroscientific basic questions that is not answered because of the limitation of the recording tools across the depth. But our grid, it is initially more focused on the intraoperative use to have a really precise submillimeter functional boundary so that the neurosurgeons can have a better determination of the healthy versus the pathological tissues and the tumor affected area. And by its large 2D coverage, we will also be able to answer some neuroscientific questions that was not answered by the previous like ECOG technologies.
So NeuroPixels, to summarize, NeuroPixels, they go deeper. So they are more in a vertical direction.
Yeah.
Just to give some ideas to our attendees. So they're recording deep areas, and they're made from different materials, silicone. They are silicone probes. So it's different. And your nanorods, they are more horizontal on a surface, so they have completely different goals and applications.
Yeah.
So, and in relation what you just said, so for mapping, for example, tumor location, why do we need your nanorods compared to imaging? So what kind of unique value this mapping brings? Neurosurgeons do MRI and go with that.
Yeah. Okay. Yeah. So MRI technology is advancing fast, and they're also like being capable of doing the high resolution MRI scan, and it's really one of the best tool that advanced the neuroscience. However, the biggest difference I could say is the sampling rate. So compared to the MRI, like magnetic imaging scanning, the ECOG recording that we're sampling data points like 20 kilo sample per second from like thousands of channels simultaneously. So for the, they're in the brain, in the many cognitive processes, they operate really fast. Like, so the MRI can miss some important information. For example, the numerical process, including the calculation. The MRI study so far was having a very much difficulties decoding the numerical process, because the numerical process in speed is faster than too fast to be captured by the MRI scan. However, with the ECOG grid, we are starting to see some interesting correlates with the numerical and calculation processes. So I would say that the sampling rate, with the increased sampling rate compared to the MRI scan, we will be able to approach many neuroscientific questions that cannot be answered by the MRI.
Yeah, unless MRI will become a cell resolution kind of imaging.
Yeah, both spatial resolution, but the temporal resolution is the most critical aspect. Yeah.
But when we need to preserve very important functional areas in tumor resection, functional mapping of boundaries is very important. Yeah. And this is where your nanogrids are unique for this kind of application. Yeah. So, because in the past, people used to lose a lot of functions with epilepsy areas resection and tumor areas resections. How about stimulation? I have a lot of questions for you. Yeah. How about stimulation? So you could stimulate for the epilepsy studies, but as I understand, these nanorods on that level of development don't have abilities for stimulation. Is it correct?
Yeah, it does not have the stimulation capability for what we afforded in this paper.
So, yeah.
Okay, so you could continue your question.
Yeah, so are you planning is it possible for like integrate stimulation, or it need to be a different technology? So are you planning to add the stimulation, or just develop, it would be interesting just to develop stimulation nanogrids, the ones who would stimulate.
Yeah. So,
because right now your nanogrids is an output recorder pretty much. So how about turning into stimulation device?
Okay. Yeah. So for this paper, we only did the passive recording. It was only for the acute, like intraoperative functional mapping, and the stimulation was just done with the clinical bipolar stimulator, or we were also able to like insert some like depth stimulator to evoke some potentials in the deep brain. However, we are targeting for the semi-chronic and chronic implantation approach, and there needs to be a way to do the electrical stimulation. So we are, with the most recent grids, we are including a stimulation site. So what we are developing is like 4,000-channel grids with like 256 stimulation channels all across the grid, so that we could stimulate like any positions on the grid. So that is happening on our side, and platinum nanorod grids have a very superior characteristics for stimulation than any other like low impedance materials such as PDOT.
So do you have real plans for developing integrating stimulation?
Yeah. Professor Daya group, together with the other groups in electrical engineering department, is developing the recording chips as well as the stimulation chip. And we are integrating that with our grid that is capable of both recording and stimulation.
It's going to be on a one platform, like on another, on the same grid, or are they going to be like beside each other?
It's going to be all integrated in the single grid. And the reason we could now do it is because this paper is reporting about a single layer device that has a like metal leads and the recording slides between the polymer layers. But now we are like scaling it up to like multiple layers, like 4 layers for the recording and 5th layer for the stimulation. So everything is getting more dense, and there are more channels and also a designated layer for the stimulation. So it's getting more like complicated, but capable of doing more things.
It's amazing. I have a couple of more questions for you. So you did this experiments for brain-machine interface. What kind of it's clear for clinical audience of nuns, clinical applications for boundaries determination and epilepsy. But how about the brain-machine interface? What kind of application? It's clear from the paper, but what kind of other additional application in the future you see with your technology?
Okay. Yeah. So, yeah, in the paper we briefly talk about the correlating the hand sensory and the motor function with the brain. So that means that our grid can essentially decode the motor intention and also could capture the sensory activities. So the Professor Dias group is also building a platform for the robotic hand with some sensory grids on it. So the robotic hands that could have a pressure sensor on the tip of it, and we will combine this research together with the semi-chronic or chronic implanted grid so that we could have a complete system, complete robotic hand that could feel and also move. So the current technologies there are many neurotech companies that is building a like implantable grids that could move the robotic hands control for the people who are physically challenged people. But we are not only going to move the hands, but we are also going to put the sensory grids on the robotic hand so that the patient can feel the touch by the electrical stimulation of the cortical surface.
So that means right now at this stage, it just recording. It might record that kind of movement of that hand, but yeah, you need to stimulate to make it move.
Yeah.
To make it feel.
Okay, so the recording grids on the patient's brain will move the robotic hands, and the robotic hands will grab the object, and they need to apply some appropriate pressure to grab the object. It
will send this feedback to the grid. Okay, I understand.
Yeah.
Great.
So that's the general direction the lab is moving toward for the brain-computer interface.
It's exciting. And my last question for today. So for epilepsy studies, what a difference between chronic intraoperative monitoring of the epilepsy. So there are a lot of ways to actually localize epilepsy area. You can do modeling. You can chronically implant intracortical electrodes and watch the pattern for a certain period of time, and which acute intraoperative. Okay. So it may be a very clinical question, but it's super interesting for all of us here. So what kind of unique value your technology brings to estimating the epilepsy area for resection?
Okay. So yeah, this maybe I would not be the best person to answer about this clinical question, but for for the epileptic like discharge and those things, as far as I know, people should undergo the semi-chronic implantation of the electrodes to actually really identify the seizure onset zone with the several like 10 minutes or 20 minutes of recording from the intraoperative setting. I heard that it is difficult to identify the seizure, so that's not enough time or enough tasks could be performed during those times. So that's why we are moving towards the semi-chronic and chronic implantation to really cure the epilepsy. But at least in the paper, we demonstrated a short acute recording to show that this grid actually captures the epileptiform activities and how it spreads. So it gives us the confidence that we could invest our time and money towards the development of this grid for semi-chronic platform.
Yeah. Thank you so much for all these answers.
Thank you.
Great discussion. So thank you for everyone who joining us today. I would like to conclude this webinar. We're going to finish a little bit earlier. So we are looking forward to see you at our 26th annual NANS meeting in January 12, 2023, and for our next June journal club on 15th of June. Thank you so much. Thank you, Dr. Cho, for coming and presenting and answering all these questions. Thank you, everyone.
Thank you.
Enjoy the rest of your day. Bye-bye.